to my dearest little champions. I hope every one of you who is watching this video is doing perfectly good. So yes, your exams are so near. You have your biology exam on 18th of March. So how far have you prepared? So is it like 60%, 80%, or is there someone with 20%? So do let me know that in the comment section, and I would definitely help you out. So this is obviously a mode of help, that is to solve model question paper, because the government has just given the question paper but not the answers. So here I am with the solution for all the questions which is there in model paper 2. So let's start without delaying any bit of your time. So over here, the part A, my dear children, consists of 1 marker question. It can be in the form of MCQ or it can be in the form of fill in the blanks. Well, the fill in the blanks portions are really easy, even the MCQs. So let's start. Select the correct sequence of events in microsporogenesis, as in you're talking about the formation of microspores. So how does it take place? So instead of going through all the options that is there, you should be having in your head what is microsporogenesis and what is the flow of it. So first thing, we have sporogenous tissue, my dear children. Now the sporogenous tissue which is there, it will be having certain microspore mother cell, and the microspore mother cell will undergo meiosis to give out microspore tetrad. They are 4 in number, they are together. Now they will slowly segregate, and that's how you get microspores. So over here, the one option that I see where it starts with sporogenous tissue is this and this. So I can eliminate these two options. Now I can go ahead and read the options, that is sporogenous tissue, microspore mother cell, microspore tetrad and microspore. This is the answer. Well, I asked you to know the flow of the content which is given even before reading the question paper, but this does not apply for all the questions. Okay, so whenever a process is being asked, at that point of time you can use your brain like this and save a lot of your time. So your answer is this. Going ahead with the next question. Statement 1 says the process of release of sperms from the seminiferous tubule is known as spermatogenesis. Is it actually known as spermatogenesis? Now if you do not read this question properly, you are gonna lose your marks. Listen. The process of release of sperms from the seminiferous tubules is not spermatogenesis. Spermatogenesis is formation of sperm. Okay, so instead of spermatogenesis, it should have been spermiation. It is what? Spermiation. That is the name of this process. Now, statement 2. So this statement is wrong. So it is incorrect. Okay. Now the spermatids are transformed into spermatozoa by the process called spermiogenesis. Is that correct? Yes, you can imagine, always imagine spermatids like kids. So they are immature, okay, most of them. And now they are transformed into the mature form, that is spermatozoa. So these ones, they are round in shape, and sperms obviously are not round, they are elongated. So for that you have to convert them into spermatozoa. And this process is known as spermiogenesis. This is entirely correct. Now that's why I'll say statement 1 is false and statement 2 is what? Correct. Let's see. Statement 1 is incorrect and statement 2 is correct. This becomes your answer. Cool. Next thing, the function of myometrium layer present in the uterine wall is 2. Now in the uterus you have different wall. Okay, the outermost is perimetrium, then you have myometrium, and the innermost is endometrium, which is the glandular layer on which the embryo will be coming, and this is the layer which provides nourishment to the embryo so that it becomes the fetus or the baby that you're looking forward for. Now over here, if you're asked about myometrium, myometrium is basically a muscular layer. M for myometrium and M for muscular layer. What type of muscles, my dear children? Smooth muscles. So as of now, I can do like this. Why? Because these muscles which are over here, my dear children, they are skeletal muscles. They can be controlled by me. But smooth muscles are involuntary. They cannot be controlled. For example, I think that basically whatever food I am having, it should not be going to my stomach. Once I have put it inside mouth, I have chewed it and I have gulped it, there is nothing that I can control. It is totally involuntary because the smooth muscles are involved up over here. So in this case also, the myometrium is made up of smooth muscles, and the muscular layer which is over here, when it contracts, that's when the baby would be coming out. Okay, now what is the use? Is it to undergo cyclic changes? No, that is endometrium. Give protection to the uterus, that is perimetrium. Exhibit strong uterine contraction during parturition, yes. Now parturition is the process during which the baby expels out of the body. Now this becomes your answer. Health and implantation process, you don't need that. Well, it is endometrium, not myometrium. So your answer is C. And why is it not marked here? But yes, it is C, okay? So you don't have, yeah, it's here. Cool. Now, dash hormone is secreted by the ovary in the later phase of pregnancy. There is only one hormone which is mentioned in your NCERT, and it said that it is being released by the ovary during the later phase. That is relaxin. It is what? Relaxin. So it's for relaxation of the muscles and everything which are over there so that the baby can easily move out. Cool? Chalo. Next question. Sperms produced by the seminiferous tubules are transported through accessory ducts. Which duct should be tied and cut for male sterilization? That means if someone is not looking for a baby, okay, so they have to opt for contraceptive methods. Now there is one method that is known as surgical method, where you are going to cut the duct which would be transporting either sperm or ovum. So in the case of males, the duct which is cut and tied, it is vas deferens, or you call it vasa deferentia. Okay, in the case of females, it is the fallopian tube, because that is the only duct that we have. So your answer for this question is what? Vas deferens, and the process is known as vasectomy. Cool? Chalo. Next question. Reasons for human population explosion are given below. That means the reason for increase in the human population. Rapid decline in maternal mortality rate. What do you mean by mortality rate? As in death rate. So decline in maternal mortality, that means less of the mothers are dying. So obviously that is a reason why there is a population increase. Okay. Now, rapid decline in infant mortality rate. Again, there's a decline, so that's a good thing again, right? So, infant mortality, as in infants are dying less. So, obviously, the population would be increasing. Now, rapid increase in death rate. If there is an increase in death rate, my dear children, don't you think, basically, it is not going to help with population explosion, but decrease the population. So, this is an odd one out, okay? Now, increase in the number of people in reproducible age. So obviously, again, if there are people in reproducible age, they will be reproducing, and again population would be increasing. So I'll say this also is an option for human population explosion. So that is being asked. So I'll say 1, 2 and 4 are the reasons for human population explosion. So hence your answer becomes B. Cool? Chalo. Next. In a dihybrid cross in pea plants, Mendel got 9:3:3:1 phenotypic ratio. It denotes that. What does it denote? In a dihybrid cross, as in you're talking about two characters at once. For example, round and yellow and wrinkled and green. So how is it two character? Basically, this is talking about the shape of the seed, and this is talking about the color of the seed, my dear children. So in a dihybrid cross in pea plants, Mendel got this particular ratio. Why? And over here, what is the ratio to suggest? 9:3:3:1 suggests that 9 of them are round and yellow, 1 of them is wrinkled and green, and 3 of them over here are new combination. It would be round, and instead of yellow, it would be green. And this would be again a new combination that will be wrinkled, and instead of green, it would be yellow. So how is this new combination possible, my dear children? Because the alleles of two genes are segregating independently. They are showing independent assortment. What do you mean by that? Basically, if I put it into two chromosomes like this. So if this is capital R, okay, and this is for wrinkled, this is for round, and this is for wrinkled. Now this is for yellow, and this is for green. Okay. Now this chromosome can go with this chromosome. Half of the chromosomes will be going. So from this pair one can go. From this pair one can go. Suppose this capital R is going, it can go with capital Y. Okay. Now this capital R can also go with small y. This small r can also go with capital Y. This small r can go with small y. So these are the gametes that you are forming. These are the gametes that you are forming. Now the gametes, when they fuse together, my dear children, obviously you get new combination. For example, this gamete from this parent, and there is a similar parent, capital R, this is one parent, okay. Now there is another parent over there having similar gametes. Suppose small r small y from that parent and capital R small y from this parent comes and fuses. Then what do you get? Capital R small r and small y. That means it is talking about round and yellow. Sorry, round and green. That is a new combination. Why did it happen? Because the alleles of a gene, my dear children, these are the alleles of one gene, these are the alleles of another gene. Alleles of two genes are segregating independently, they are separating independently. So your answer becomes this. Cool? Yes. Children, are you enjoying? Are you understanding? Do let me know in the comment section, okay? Now, question number 8. A DNA segment has a total of 1000 nucleotides. Please read this question really carefully. Okay, how many nucleotides are there? 1000 nucleotides are there, my dear children, out of which 240 of them is adenine, that is adenine, okay, containing nucleotides. How many pyrimidine bases this particular DNA segment possesses? That means you're asked about how many T and C nucleotides are there. So if you know about adenine is 240, that means you also know about thymine, that is the counterpart of it. So if adenine base pairs with thymine, right? So this also will be 240. This also will be 240, my dear children. So 240, 240, fine. Now the common mistake we do is we minus 1000 minus 240. We do this for finding G. But my dear little children, how many nucleotides do you have in total? You have 1000. So 240 plus 240, basically 480 of the nucleotides we have already counted in this particular 1000. Now we have to count the rest of them. So 1000 minus 480 is what? 1000 minus 480 is basically around, okay, 520. It is what? 520. Now 520 nucleotides are still left. Now 520 again you have to divide between G and C. So 520 divided by 2, right? So what do you get? Again, over here for everyone, sorry, oh my god, for everyone who might get confused, it is 260. Okay, what do you get? 260, my dear children. So this is also 260 and this would also be 260. Now if you do total, my dear children, it will be 1000. So you are asked how many pyrimidines are there. So T and C, how many of them are there? So 240 plus 260 is equal to what? 500. So this becomes your answer. Cool? Works? So the calculations would just to confirm, otherwise what if I do a mistake? So yes, next. P plus Q whole square is equals to P square plus 2PQ plus Q square represent an equation used in what? Population genetics, molecular genetics? Just a second. Yes. Mendelian genetics or biometrics? What is the answer for this? Cool children. What does this particular thing represent? Well, this is Hardy-Weinberg's principle, according to which alleles in a population is always same. That is what he said, generation after generation. So this is for a population genetics, my dear children. This is for calculation of population genetics, a straight question. So there is nothing to think about it over here. Okay. Now, a farmer working in a field was bitten by poisonous snake. Doctor gave him an anti-venom treatment that contained preformed antibodies. So a snake was there, it bit a farmer, and the farmer went to the doctor, and the doctor is giving him anti-venom. And what does this anti-venom contain? Preformed antibodies, as in ready-made antibodies. This type of immunization is known as where our body is not producing antibodies. We are depending on outside source. Someone else is giving us ready-made antibodies. This is where our body is not actively participating. So it cannot be active immunization. It is passive immunization, my dear children. What is it? Passive immunization. Preformed antibodies are given to us. Now suppose our body is producing it. That's when it is active immunity. But do you think it can produce in one go? For the first time when snake came and bit us, our body would not be producing too much of antibodies in the first go. Second time if the same thing happens, probably it might have a memory of it, and because they have memory cells in our body, which is why again obviously a large amount of antibodies would be released. But as of now, when snake will come and bite, we have to take preformed antibodies. There is no other option to be safe. Okay, because these toxins work really fast, we could be killed, so we don't want that, right? So passive immunization is best for that. Even your tetanus enzyme, I said exam, because the exam thing is too much into our brain also. It feels like we are giving exam instead of you. I don't know how prepared are you. I definitely know how prepared am I. So my dear children, please do your preparation really well and cheer up. It's just an exam. Do not go thinking that it's exam, I will lose my marks, what will I do, my future. My dear children, just be happy like this. Okay, study, give your maximum, give your 101% and see the change. That's what I want to say. Okay, now an agricultural labor was spraying some powder mixed with water onto fruit trees to get rid of insect larvae. Okay, He put some powder into water and he sprayed. Now, which of the following biocontrol agent could have been used there? Biocontrol agent as in, for example, we put chemicals and all and to kill the pest, but apparently it kills the plants also. So, we don't want that. Instead, we are depending on organisms that could be eating these pests. They could be our best friends, right? So, what is this powder sort of thing that is going to kill the insect larvae? So, over here Bacillus thuringiensis is there, Trichoderma, dragonflies and ladybird is there. Now, listen. First thing first, dragonflies for mosquito larvae, ladybird kills basically aphids which is there. Now, did I say trichoderma? Sorry, dragonfly is for mosquito and ladybird is for aphids, and trichoderma which is there, it is in the roots and it kills the other root fungus which is not really good for the plants. Now, Bacillus thuringiensis is the perfect answer for this because this is the one that would be infecting the insect larvae. They are going to release Bt toxin, okay, which is inactive in their body, but when these Bacillus thuringiensis are eaten up by the insects, in the insect body gut, in the gut there's an alkaline pH, so the toxin becomes active and basically cleaves the epithelial cells around there and totally damages it. So this becomes your answer. Cool? Yes. Next, choose the correct sequence of polymerase chain reaction steps from the following. Polymerase chain reaction is their only motive is to what? Amplify or increase the number of DNA or replicate the DNA, right? So for that, first thing that you have to do is, this is suppose a DNA. This DNA would be bound to each other. Now this, you have to break it open, right? So this process is known as denaturation. After denaturation, what do? You want to make a copy, so for that you need DNA polymerase, but DNA polymerase does not start off like that. It needs certain primers over here, certain sequences which should be already there. Now these primers are added and this process of adding primers is known as annealing. Later on, in the next process, my dear children, DNA polymerase will come and extend this in both the direction. This is known as extension. So in that way, you're getting from one DNA, you're getting one DNA and second DNA as well, 2 DNA. So this becomes your answer, my dear little children. Okay, going ahead. Use of bioresources by multinational companies and other organizations without proper authorization and compensatory payment is referred to as. So, I am using some countries' bioresources, okay, and I am not telling them, I am not giving them money also, I am just using that. Now, what do you call this? This is known as biopiracy. This is known as biopiracy, my dear children. Chalo. Next. An example of ex situ conservation is, as in conservation outside their natural resource is ex situ conservation. Now, you have national parks, sacred groves, biosphere reserves and zoological parks. Zoological park is basically zoo. So, do you go to a forest to see animals in the town and not city and things like that? No, you go to zoo. In zoo, my dear children, the animals from the natural habitat are being put over here. So, it is ex situ conservation. They are not preserved in their natural habitat. If you have gone to Wayanad and all, you might be saying that there is biosphere reserves. Sorry, there are so many reserve forests, tiger reserves and all. Basically, that's their natural habitat. We have built a road in there. You get it? So, that is in situ conservation, but this is what? Ex situ conservation. So, your answer is what? Zoological parks. Okay, sacred groves is basically when, I'll tell you because that is the only option that might confuse you if at all it's there. Now, sacred groves is basically, I have a region, I have some religious, emotions attached to that particular region, I want to safeguard it. Okay, but in that way, when I am safeguarding that particular region, am I not saving the plants over there, the animals over there? So, again, that becomes in situ conservation. So, the answer for this is zoological parks. Going ahead. The graph given below shows species area relationships. Which of the following equation correctly represent the curve? So, you have B is equals to log S is equals to, I am directly going ahead with the answer. So, A is equals to S is equals to CA where the power is Z, and then B is log S is equals to log C plus Z log A, and then C is A is equals to CSZ, and then D is log Z is equals to log C plus S log A. Well, what does this type of curve represent? So, this is about the species richness, my dear children, and the most apt answer for this is B, as I already said. Okay, this would be the answer, B option. Cool? Now, going ahead, just give me a tiny second. Okay, where is it? Okay. So we have certain fill in the blanks question. Let's solve it. So first thing, statutory ban on dash is required to check increasing female feticides. First of all, my dear children, this is the most scoring region in your paper. Pakka 5 marks you will get. Okay, so these are the options given below and you have to choose from them. So statutory ban is required to check increasing female feticides, as in people are killing female children. So something was banned. What was that something? It is amniocentesis, where what was done, you know, babies know they are covered in an amniotic fluid. So an amniotic fluid would be having certain cells of this baby only. When the baby is there, it will shed some cells into this amniotic fluid. So these cells are being taken up and then the chromosome is checked, and if it's a female child, people usually kill it. But was amniocentesis used for this? No. It was for finding genetic abnormalities, but people are using it for other causes. Now you should not be someone like that, okay? So yes, the answer is amniocentesis. Going ahead with the second option. Second question. A fish thought to be extinct and caught in South Africa in 1938 is. So it was thought to be extinct but again was caught in South Africa, my dear children. This is a very famous fish and it is thought to be the one that basically evolutionized to form the amphibians, that is the ones that live on land as well as water. For example, frogs, salamanders, everyone. So that connected link is, my dear children, lob fins. Okay, we have lob fins, or the other name for this is coelacanth. Fine. So the first answer is this, the second answer is this. Now the genus of mycorrhiza forming fungi is, so you know mycorrhiza is a combination, symbiotic association of plant and the fungi. So the fungi belongs to which genus, my dear children? It belongs to glomus. It belongs to glomus. This is the answer. Now next, autonomously replicating circular extrachromosomal DNA of bacteria is known as, it is autonomously replicating circular extrachromosomal DNA, my dear children. So there is only one thing, that is plasmid, that does that. Okay, so your answer becomes plasmid. Next thing, rate of formation of new organic matter by consumers is referred to as. So rate of formation of new organic matter, not the one that they got from the producers. So I am suppose a rabbit and I am eating the plant. Now whatever food I have for the other organisms that would come and eat me, this production of new organic matter by me is known as secondary productivity, not primary. Primary is done by plants. This is known as secondary productivity. So if this, okay. So this was the fourth one and this is the fifth one. There is no primary productivity. Don't go for that trap. Yes. And you saw how fast we did the MCQs, right? You know, you need the same type of thing for KCET examination also. You should be having really good practice for MCQs, and here we are with the Abhyas KCET test series, my dear children, where you would be getting 35 full syllabus and chapter wise tests, which you should be definitely not missing. Obviously, why should you miss when the teachers whom you already know are there for teaching you? And also there will be detailed video solutions for any kind of problems which is there, so you will not be facing any problem. Okay, there will be chapter wise one shot revision. There will be, you know, past 5-year paper has been analyzed for making up this thing, so do not worry about anything. We are gonna get it covered, and it would be for rupees 1499, where 1 rupee is been added for getting PU2 test series as well. So if you are someone who is in PU2, right, that's why you're watching this video. So if you're interested in giving KCET as well, then definitely you should be doing it, and it will be really good. In that way, we are gonna prepare, practice and perform. Okay, going ahead, let's perform part B questions, which is 2 marker questions, my dear children. You have to attempt any 5 questions, but obviously over here we have to attempt all of them. So differentiate between geitonogamy and xenogamy. What do you mean by geitonogamy and xenogamy? So it's an easy question. So over here, suppose there is a flower, there is another flower. Over here I have the female part as well as the male part. Suppose the flower is like this. From here the pollen grains are landing up to the next flower, but it's on the same plant. Okay, or probably suppose there is only one sex in one plant, another sex in another plant. So what will happen? Basically, from here the pollen grains would be traveling there. So whenever it is happening between different flowers of the same plant, it is known as geitonogamy. So if you see, it is like autogamy. It's a self-pollination. It's genetically, it's like self-pollination. Otherwise, it is like cross-pollination. Genetically, if we see the same plant, so it is like what's say autogamy, but obviously it is a form of cross-pollination, my dear children. Now what do you mean by xenogamy? Xenogamy has another thing to say. So suppose this is a plant and this is a plant. Again you have flowers, some female flowers up over here and some male flowers up over here. Here to here things are going to come, but here two separate parent plants are being involved. That means two separate genetic material as well. Now this is known as xenogamy, which in true sense is cross-pollination. So geitonogamy is a transfer of pollen grains from the anther of one flower to stigma of another flower of the same plant, genetically similar to the parent plant. Agree? So this is next point. Okay. Now here transfer of pollen grains from anther to the stigma of different plant, genetically different from the parent plant, and this brings out variation. Over here, do you find variation? That could be another point. No much variation are gonna be seen over here. Okay, because the same plant only, right? Now next question. Write a short note on sex determination in birds. In birds, they have a unique way of sex determination, my dear children. I would be just explaining with the help of this. So in birds, they have ZZ and ZW, what say, sex chromosomes. Suppose it's ZZ, then it is to be a male, and suppose it's ZW, it will be a female. Cool. Now over here, the gametes are Z and Z. Okay, over here the gametes are Z and W. Now let me tell you, this is an example of female heterogamety, the only example of female heterogamety. That means females are producing two type of gametes. Usually male does that. Okay, so this is an example. And suppose this and this comes together, you get ZZ, that is a male. This and this comes together, ZW, that is a female. This and this again a male. This and this again a female. So 50% chance of a male, 50% chance of a female, and that's how sex determination works in the case of birds. And this is an example of what? Female heterogamety. Cool? Chalo. So are you? Yes. So two different types of gametes in terms of the sex chromosomes are produced by the female, that is female heterogamety. In this type, female is heterogametic, ZW, whereas the male is homogamitic, that is ZZ. Homogamitic is the same type of gametes are being produced. Now, the female produces two type of eggs carrying Z and W chromosome, whereas the male produces only one type of sperm, that is carrying Z chromosome. If sperm carrying Z chromosome fertilizes with the egg carrying Z chromosome, then zygote will develop into male. And if the sperm carrying Z chromosome fertilizes with the egg carrying W chromosome, it will be a female, just like how I explained. Now over here, you do not have to write about autosomes and all. Okay, don't have to write about autosomes, just write ZZ and ZW, and that will be more than enough. Make sure you're writing parents, then you're writing gametes, and then you're writing F1 generation. Okay, please make sure like that, and you have to cross it with proper lines. Don't do otherwise. Okay, that's all clear. Chalo. Next, mention the levels where gene expression can be regulated in eukaryotes. As in, if I want to allow expression of gene in eukaryotes, in what all ways can I stop it, or can I basically enhance it? So first thing, transcriptional level, I can maybe, if I do not want, suppose I do not want. So in transcription level only, when the DNA is going to get transcribed into mRNA, I can bring about some changes over here. I would not allow this process to happen. So this is one way. Okay, second thing, processing level. You know, the DNA is not directly making mRNA, it forms hnRNA which gets processed to mRNA. So in this processing, I would not do the processing. In that case also, I can stop the whole thing. Okay, now transport of mRNA from the nucleus to the cytoplasm. To make proteins, no, this particular mRNA which is there in the nucleus has to go out. Now I can stop it from going. Again, I can stop the whole process. Now translational level, as in when the mRNA is finally outside, still I do not want tRNA to come bind over here and the process of translation to happen. At this level also, I can basically stop it. Okay, so these are the different levels in which I can regulate, gene expression in the case of eukaryotes. What about prokaryotes? Prokaryotes, mostly it is only the initiation of transcription where it can be controlled. Okay, which is why they have operators, you know, lac operon and all. So they have operators. On the operators, if some repressor protein comes and binds, then obviously the process of transcription would not be happening. So people who are worried listening to my answers, I want to tell you something. I get you that you might not have covered everything. Your friends whom you talk to might have covered a lot. Listen, I also have been in the same phase. Okay, it's not like you are the only person on earth who have went through all this thing. It's fine. But if any one of us just sits thinking that and do not work on it, we are not gonna win this game. So we have to win this game, right? So please overcome that and come over here. If you do not find an answer that you are understanding, mark it, right? It's 23rd question I didn't understand, I have to revisit, I have to go through my NCERT, I have to understand, then I have to come. This model question paper is really important, which is why we have making videos on it. Otherwise, why should we make video on it? You get it? So these are long and the views might be very less. The views are not very much, right? But still we are making. Why? Because we think that it is important for you guys. So please consider this to be important. Revisit the question in your NCERT only. You can go read through your NCERT, understand that, and then it will be fine. But you should not leave it. You did not leave the question. Now write a short note on Neanderthal man. You know human beings, how do they came? So first we had Dryopithecus and Ramapithecus, okay. Dryopithecus were more ape-like and Ramapithecus were more human-like. Now from them came Australopithecus, from them came Homo habilis, from them came Homo erectus, and from them came Neanderthal man. Now Neanderthal man were towards like more closer to us. So they were found about 1 lakh to 70,000 years ago, my dear children, and the brain capacity, cranial capacity was 1,400. Was it? What is it? 1,400. And they were starting protecting their body. They covered their body, and they were the ones who buried their dead also. So that is something uncommon. So they were kind of nearer to us. When this was happening, parallelly, something else was also happening. Homo sapiens developed. That is basically the present day us that we have. The modern human beings. Homo sapiens also developed throughout. Okay? In the middle of these years. So yes. So Neanderthal man, again, if you just study like this, you won't be able to remember. Relate and learn, it becomes more easy. Neanderthal, so 1 lakh to 70,000 years ago, 1,400 cc cranial capacity brain was there. They protected their body and they started burying their dead. They had some emotions coming up there. Okay? So yes. Now, draw the structure of an antibody molecule. Most repeated question, my dear children. So many times this particular diagram has been repeated. So over here you have to realize you have to make what? Heavy chain, first thing. Okay? You have to make heavy chain, and also you should be making light chain. So your light chain looks like this, heavy chain looks like this. In both the regions you would be having, sorry, I have made a cut over here. So there is no cut, okay, just a second. So this region that you see, my dear children, it is variable region. It is what? Variable region. It differs in different antibodies. You know, there is IgG, A, M, E, D. There are 5 antibodies. All of them, they have different variable region. The other region that you see over here, from here to here, it is known as constant region. Now this is heavy chain, this is light chain. How many heavy chains do you have? You have 2. So you have to write H2. Now how many light chains you have? You have 2. So you write H2L2. Okay. Now over here you have a site for antigen binding. So you write it antigen binding site. So I'm not making 2 arms because it is just there. Now between the 2 heavy chains you have so many disulfide bonds, my dear children, as you can see over here. So they have in between them and also within them. Okay. Now here also between the light and heavy chain also there is one disulfide bond as well. So this is the structure you need to draw. You need to know how to draw it. Cool. Chalo. Next, write the functions of genes Cry1AB and Cry2AB. I told you about Bacillus thuringiensis, that is BT. Now BT is able to produce a toxin that is a protein basically. Why? Because it has certain genes for it. So the genes are Cry1AC, Cry2AB and Cry1AB. Okay. Now Cry1AC and Cry2AB, both of them are responsible for killing cotton bollworms. Now they are specific. They produce toxins that kill cotton bollworms. Cry1AB is the one that kills corn borers. That kills what? Corn borers, my dear children. So how do I remember it? Okay, so first letter starts with C. First letter starts with C. Second letter starts with B. Second letter starts with the cotton bollworm. So you see corn borers also just like that only, ma'am. But cotton bollworms is kind of long, and here also 2 cry genes are involved. This is also long. Now Cry1AB, the first letter C and the last letter borers. Okay. So over here, the genes you're not asked about this one, you're asked about this and this, so you have to tell. Cry 1AB is for corn borers, okay, and cry 2AB is for cotton bollworms. That is what is being written over here as well. So they produce this protein which is toxic to corn borer. It is used in genetically modified crops like BT cotton, BT corn. So what do you do is that you put the gene inside the plant. So plants will also be producing this toxin. So no another pest will be coming and eating it. You get it? That is the level. Got it? So yes. Now briefly explain the significance of David Tillman's long-term ecosystem experiment using outdoor plots. So, his research has shown that greater plant species diversity tends to increase ecosystem productivity, stability and resilience. As in, if you have more diversity, it will lead to what? More stability. That is what he said. So, by manipulating species composition in these plots and observing the effect over time, Tillman was able to demonstrate that diverse ecosystems are more capable. So, he tried putting out certain species. He tried inserting some other species. He saw what change does it make? So he saw that basically the ecosystem which had more diversity were capable of maintaining stable functions such as nutrient cycling and carbon storage in the face of environmental stresses as well. These experiments have been foundational in understanding the relationship between biodiversity and the ecosystem services as well. This is a straight question, okay? Nothing much to say, you just have to use little bit of your own things also. It's like writing English answers, in where you write your own feelings, right? Similarly, you have to put on something, but yeah, have the main part over there. Cool? Now, draw a LS of, okay, I just messed up, I think, just a second, okay? Did I not mention 3 marker? So, I assume from this point onwards, it's 3 marker, okay? From this point onwards, it's 3 marker questions which are there. Again, you have to attempt any 5 of them. So, draw a LS of grass embryo diagram and label the following parts: scutellum, coleoptile, shoot apex, radicle, root cap and coleorhiza, my dear children. So, over here everything is being mentioned, but you need to have a basic understanding of this. Now, grass embryo, as in you are talking about monocots. That means they just have 1 cotyledon. What about dicots? They have 2 cotyledons, my dear children. Example of monocots is maize, your rice and wheat and everything. Okay, they cannot be broken down into 2. Now, listen, monocots. So, usually endosperm is there. You know endosperm, right? Endosperm provides nourishment to zygote, which helps it convert into embryo. Now, this embryo says that yes, I have used all the endosperm and that's how I found myself. But instead of having an endosperm, I will have cotyledon of my own which will provide me nourishment and everything. So, I am utilizing all the endosperm to form the cotyledon. But in the case of monocot embryo, they are still leaving back some amount of endosperm, which is why they just have 1 cotyledon. They are not making 2 cotyledons. So, now in an embryo, there will be an axis, embryonal axis, an area which would be giving rise to the shoot, that is known as shoot apex or plumule, an area that will be giving rise to the root, that is known as radicle. Now, this radicle is covered by root cap. Okay. Now, over here, my dear children, here is your cotyledon, and the name of this cotyledon is scutellum. Since there is only 1, so they thought of naming it, that is scutellum. And there will be a little amount of endosperm also left behind, okay, which will provide nourishment. Now, the plumule says, I am feeling alone, I also want some friend. So, basically, there are certain layers of friends, and this is known as coleoptile, which will be giving rise to, let's say, leaf and everything, okay, leaf primordia. Now, here also radicle also feels the same, so they also have a friend, and that is known as coleorhiza. So, that is the story of the embryo, grass embryo, and the same structure is being designed like this and given to you in your NCERT. So, this thing that you see is the scutellum adhesion, that is the cotyledon. Endosperm is not being mentioned over here. Now, you have the coleoptile covering the shoot apex. So, this is a shoot apex which is covered by the coleoptile region. Here, radicle is there covered by root cap and then coleorhiza, and then there is epiblast as well. So, this is the structure of the LS of the grass embryo. Cool. Understand the basic, then you will be remembering each and every part. Okay, even if you are asked to label, you can label it properly, right? So, yes, please have the practice of this particular drawing. It's very, very, very important. Going ahead. In the figure given below, parts A and B show the level of hormones which influence the menstrual cycle. Study the figure and answer the questions that follows. Name the organs and glands which secrete the hormones represented in label part A and B. So A and B is there. So you have to name the organs and the glands which secrete the hormones. So here estrogen and progesterone has been mentioned, my dear children. It is produced by ovary, the female primary sex organ. What about FSH and LH, that is follicle stimulating hormone and luteinizing hormone? It has been secreted by pituitary gland. What gland? Pituitary gland. And who tells them to release all this thing? It is hypothalamus. Hypothalamus, the bap ka bap. Okay, so it's like grandfather of all the endocrine organs which are there in the body. Now, state the role of hormones secreted from part B on the uterus of human female during the menstrual cycle. So part B, you have estrogen and progesterone. So what is the use of it? So first thing, you have different phases. You have menstrual phase, that is when the flow happens, that is from 1 to 5th day. Then you have a proliferative phase where the endometrium will shut it downwards to slowly grow back. Thinking that again, I'll wait. What if the egg is released and fertilization happens? So there is a reformation happening. So the estrogen is there, which is responsible for this proliferation slowly. Okay. Now when it reaches the 14th day, my dear children, the graphene follicle which is there, it ruptures to release the egg finally. Now at this point of time, obviously fertilization can happen, cannot happen. So when the graphene follicle ruptures, it leaves back something that is known as corpus luteum. Okay, what does this leave back? Corpus luteum. And this corpus luteum, my dear children, releases progesterone in more amount. Releases progesterone and estrogen in more amount, such that now the progesterone will make the endometrium line more thickened, such that what if fertilization will happen? Okay, cool. And now what happens? The progesterone which is there, which is released by the corpus luteum. And who maintains the corpus luteum? It is the LH that maintains the corpus luteum. Okay. So basically ovulation is caused by the LH. Whenever there is LH surge, when LH increases way too much, that's when graphene follicle ruptures and the corpus luteum is formed. So LH is the one that maintains it, and it releases progesterone and estrogen. This progesterone has one problem. It goes and tells the hypothalamus that, hey, stop the secretion of LH and everything. I don't want to see them. So this guy stops secreting. Then what will happen? The corpus luteum will not be formed. And then the progesterone will also not be formed. It's because it took the decision in very much haste. It was very angry with LH, that's why it did it. But it's totally wrong. Now, what will happen? There is no progesterone, and that's when again the menstrual cycle happens. That is, there will be a flow. Okay, endometrium shuts down again. If fertilization happened, my dear children, this corpus luteum is still being maintained. Even though this projection goes and says that still the corpus luteum is maintained by other hormones. Okay, that is HCG, human chorionic gonadotropin. So state the role of hormones secreted from part B. I hope you got it, but let's see. So part A is pituitary gland and B is ovary. Endometrium of the uterus regenerates through proliferation. Okay, that is the role of hormones secreted from part B on the uterus on the human female during menstrual cycle. So instead of writing that, you can write in phases, as in the proliferative phase, estrogen regenerates the endometrium. Okay. Now in the secretory phase, that is the next phase that is there. Secretory phase, progesterone is being released by the corpus luteum, and it maintains and thickens the endometrium. It what? Maintains and thickens the endometrium. And along with estrogen, okay, estrogen is also doing the same work. But progesterone does it more during the next phase. Cool? So yes. Next question. Suggest the 3 simple principles through which we can prevent sexually transmitted diseases. First thing, practice safe sex. That is, use condom. Condom is the best thing because it is acting as a physical barrier for all the sexually transmitted diseases. Sexually transmitted diseases are transmitted with the help of fluids, genital fluids, right? So when using a condom, there is no chance the genital fluids are mixed, and that's why you can prevent sexual diseases. Now, apart from that, maintaining monogamous relationship is also very much important, because the higher chance you are basically having sexual contact with multiple person, that means there are higher chances of developing STDs as well. So in monogamous relationship, when you have a chosen person over there, you know their diseases and everything, and that becomes more safer. Now apart from that, there is one thing which has not much things to do with the sexually transmitted diseases, but yes, avoid sharing needles. Now, As in sexually transmitted diseases, my dear children, they can be spread through body fluids. Okay, they can be spread through body fluids, which includes blood as well. Now, which is why it is said that you should not be sharing needles or syringes. So, HIV is a sexually transmitted, hepatitis is a sexually transmitted disease which can be spread through needles as in blood as well. So, instead of writing this, because since they are asking about prevention, now there is one point given in your NCERT, that is talk to a counselor, but that is, yes, you can talk to a counselor to prevent it, but that is not absolutely a way, but yeah, you can write that as well. Cool. But if you write this, teacher might get confused, so you have to write it properly, because since STDs are also transmitted through body fluids like blood, which is why avoid sharing needles is also very much important thing. Otherwise, yes, obviously, go seek help of a counselor is the best way. Okay, fine. Next. Now, what is adaptive radiation? Give any two examples. So, adaptive radiation is a very fun thing. What happens, I'll tell you, and then I'll explain about all this thing. So, suppose this is the area that I have, and these are the organisms that I had initially. Now, few of them went to this area. Suppose these were Darwin's finches. So, Darwin was the person who found that. So, Darwin finches, no, he saw that few of them went to this area and they were feeding on insects. Few of them went to this area, they were feeding on seeds. Here they went, they were feeding on grass. Basically, what happens is that here, when Darwin's finches were there, they had mutations in their population. So, suppose two of them which had the mutation, they went along different area, and they saw that I can eat insect very well. Why not I stay here? So, they evolved to have a beak like that. Now, there were other organisms which other mutations, they didn't know that they were good at eating seed, but when they went there, they got to know it, and they evolved to have children who also had the same type of beak, and they stood there. So, over here, if we see from one common ancestor that they are having, my dear children, one common ancestor that they are having, they are moving around to different areas, geographical areas, and they are going and habitating over there. And this is what adaptive radiation is. You are radiating and you are getting adapted over there. Now, example, if you are asked, you have two examples. One is of Australian marsupials. Australian marsupials, they also went like that, and similarly in the same area you had them placental mammals. Both of them they moved to different areas, and for example, Australian marsupial, one guy came here, placental mammals also came over here. Obviously they are in the same habitat, so they were kind of having the same structure as well. Okay, they were having, but they do not have the common ancestor. They are different organisms. So, this is an example of convergent evolution as well. In convergent evolution, what happens? Basically, two different organisms come to same habitat and have same functions. Similarly, these guys were an example of convergent evolution, just saying, because you can get a question for that as well. Now, so this is what you have to write, any two examples, you can go read it. So, during Darwin's journey to Galapagos Island, that's when he observed small black birds called Darwin's finches. The finches were diverse in their food habitats, like original from seed-eating features to many other fawns, with altered beaks arose, enabling them to become insectivorous and also vegetarian finches. Now, adaptive radiation in marsupials. Number of marsupials, each different from other, evolved from ancestral stock, but all within the Australian continent. For example, Tasmanian wolf, tiger cat, marsupial rat, kangaroo, all of them from a common ancestor, my dear children. Similarly, placental mammals also. In Australia, adaptive radiation is exhibited where placental mammals are evolved into varieties of which appear to be similar to corresponding marsupials which are there. I told you, in the same habitat they are coming. So, placental wolf is there for Tasmanian wolf. Okay, so they both are basically having the same character. So, this becomes your answer for adaptive radiation. Okay, going ahead with the next question. Name of the drugs, its source and nature is given in the table below. Find A, B and C. So, A is dash. So, name of the drug is dash and it is obtained from Papaver somniferum and it is acting as a depressant. My dear children, there is only one drug that acts as a depressant from Papaver somniferum, and that is heroin. That is heroin. The other drug obtained from this particular plant is morphine, and morphine is going to be used like a painkiller and sedative. Okay, it is used by doctors while operations. Okay, that is morphine. Now, cannabinoids are there. Cannabinoids are being produced from the source plant. What is the name of the source plant? It is Cannabis sativa. It is cannabis sativa and it has effects on cardiovascular system. Let me tell you, in three different drug groups, no, only one is going to affect the cardiovascular system according to your book. Both of the others are going to affect the CNS, central nervous system, and that answers your next question. Cocaine, which is obtained from Erythrosalem cocoa, is going to affect what? The central nervous system. So over here, Papaver somniferum, the drugs which are there produce heroin and morphine, are going to affect CNS as well as gastrointestinal tract. Okay, cannabinoids are going to affect the cardiovascular system. Okay, and cocaine is also again going to affect the CNS. So your answer becomes heroin, cannabis sativa, and effects on central nervous system. Central nervous system, that means your brain, your spinal cord, everything. Okay. Now, how did an American company, Eli Lilly, use the knowledge of rDNA technology to produce human insulin? So, I'll tell you the story. Are you ready for the story? So, again, in human insulin, my dear children, you have proinsulin version where they have A, B and C polypeptide chains. Now, the problem is that in our body, when it is produced like this, the C peptide is being removed. A and B are joined, and that's when you get the actual version of insulin. Okay. Now, if I put this gene directly, okay, ABC producing gene directly in an organism and I wanted to produce, it will still produce a pro-insulin version. But do they have enzymes which will cut C down and give you insulin? No. So, Eli Lilly came with the knowledge that what if we are separating the genes also. Let's separate A gene and B gene. We are going to put A gene into E. coli with the help of a plasmid vector. Similarly, we would be putting B gene into another E. coli with a plasmid vector. So, now both of them are producing A and B genes. So, okay, both of them have A and B genes, and A gene will be producing whatever protein is needed, B gene will be producing whatever protein is needed. Now, both of these proteins which are there can be taken out and can be combined to give what, my dear children? Insulin. So, why was this drastic step being taken? Because people were initially using the insulin extracted from pigs and all. Now, even though we are mammals, they are mammals, the thing is that our body is obviously different from them, and some people have allergy to animal resources. So, that is why, what if? Let's do this. Let's take the human gene and make a copy of it and just put it there. That is the best. And that's how things worked. So, this is what was done by Eli Lilly. Now, yes, the answer is isolation of insulin gene. The gene for human insulin was isolated from human cells. Insulin is made up of two polypeptide chains, A and B. Using recombinant DNA technology, the genes for A chain and B chain of insulin were separately inserted in plasmid vectors. Okay, the plasmid, this is the self-replicating DNA molecules. Okay, now what was done? The recombinant plasmid carrying the insulin gene were introduced into Escherichia coli, that is E. coli, and production of the insulin chains. The bacteria were cultured in large bioreactors where they produced A chain and B chain separately under controlled environment. Later, purification and assembly was done, but the two polypeptide chains were extracted, purified and combined, and that's how you got the human insulin. And Eli Lilly is still there. It's a very famous company. I hope you might know it. Okay, so yes, this is the answer. Now, 34th question. An example for grazing food chain is given below, that is grass eaten by grasshopper, grasshopper eaten by birds, and birds eaten by man. Now, graphically represent this food chain through pyramid of energy and write different trophic levels with their energy content. Chalo, let's make it. So, obviously, pyramid of energy is always, my dear children, upright. It is not inverted. So, over here, what do we have? We have trophic level 1, that is occupied by grass. Trophic level 2 occupied by grasshopper. Okay. Trophic level 3 occupied by birds. And trophic level 4 occupied by man. Cool. So, the sun's energy, if you remember, okay, almost 1 to 2% of the sun's solar energy is being taken up by grass. I mean the visible light, I'm talking about that. Okay, that is the producers. Suppose it has 10,000 kilojoules of energy. Now, what is the energy transferred to the grasshopper, my dear children? Only 10%. Only 10% of 10,000 will be transferred. So it will be what? Basically, you're talking about 1000 kilojoules. For people who do not know, I'll tell you how to take 10%. So 10,000 into 10 divided by 100 is how you calculate it. So you get 1000. Make less, 10 less. Again, obviously, why is this happening? Why is only 10% given? Because 90% of it can be utilized by the organism itself, and also when transformation of energy happens from one trophic level to another, there will be some loss as well. So this is the reason. Now from here to here, again 100 kilojoules, because you are just giving 10, and from here to here again 10 kilojoules of energy. Which is why you cannot have more trophic levels. Or you do not have anything to give, what will you give? That's why in an ecosystem the trophic level is completely reduced. Okay, it will be 4 to 5, that is the maximum it can go. So this is the energy pyramid. Okay, chalo. Going ahead to the next question. Draw a neatly labeled diagram for sectional view of mammary gland. So I'll tell you how, again, the way to remember it, that will be much better. So my dear children, if you see the mammary gland over here, it has lobes. So this is one lobe, this is second lobe. So you have multiple lobes over here. Okay. So how many lobes are there? Mammary lobes, almost 15 to 20 mammary lobes are there. And the mammary lobes have so many small structures like alveoli. Okay. So mammary lobes will be opening into alveoli, or basically mammary lobes have lots of alveoli, and the alveoli are going to open into mammary tubules. Mammary tubules, which is not shown in the diagram. I'll tell you why. Because if you see, if you see, this is the structure, lobes with alveoli. Behind the alveoli there will be tubules, my dear children. For example, this and all is a tubule. Tubules are going to open in a duct. So this thing that you see over here, this is a duct, this is a duct. So these are mammary ducts. Now duct will be coming together to form ampulla, this enlarged region. Now the ampulla is going to come together to form lactiferous duct, and then here ending with nipple, and this is the areola which is there, and the whole breast is covered by lots of fat. Okay, and behind it you have ribs, all the muscles between the ribs and everything is there. So if you understand the concept logically, obviously you will be able to remember it better. So mammary lobes, inside that you have trees. Just make trees with multiple branches. So there should be lactiferous duct, it should be branching out to a broader thing, that is ampulla. Ampulla should be branching out to different duct. Duct should be further branching to form tubules. Tubules should be making what? Tree-like structures which are mentioned over here. And then above it you should be making mammary lobes as well, and towards the side you should be making fat. Got it? So that is the structure of mammary gland. Now schematically represent the inheritance of flower color in snapdragon and draw conclusion. So in snapdragon what happened is that so instead of making the diagram you just have to mention the things. So red color flower were taken, my dear children. Okay. And the genotype was capital R, and they were fused with white color flowers. Now as you can see, capital R is a dominant trait and small r is a recessive trait. That means red is a dominant trait and small r, that is white, is a recessive trait. When they were fused, so they have not shown the fusion, which is why I have to show it. Why is it happening? Yes. So capital R, small r, these are the parents. Don't go write P and come, because just ma'am said, right, parents, okay. Now over here you have gametes, which is capital R, small r. Now you fuse the gametes, you get capital R, small r, again capital R, small r. If you see, all of them has one capital R, and it should be dominant over this, but is it? No. Which is why all of them, my dear children, will be pink in color. Will be what? Pink in color, which is not like red. It is not completely dominant. So they are going to show what? Incomplete dominance. They are going to show incomplete dominance. Tell that in your answer. Okay, in F1 generation, you can see that all the flowers are pink in color because R is not completely dominant over small r. Okay, it is incomplete dominance. Now what they do is that they took the pink flowers. Okay, let's do much more experiment. And now again, same things were repeated. Capital R, small r, and capital R, small r. You have the gametes capital R and then small r. Capital R and then small r. From here to here, if you cross, you get capital R, capital R. This and this, you get capital R, small r. This and this, you get capital R, small r. This and this, you get small r, small r. So if you see, my dear children, okay, so this is red, this is white, because there are two alleles. Here also there are two alleles, so there is no point of incomplete dominance. But here still they are showing incomplete dominance, which is why they are pink in color. If you are asked the phenotypic ratio, only one is red, two of them are pink and one is white. If you are asked about genotypic ratio, one is capital R, capital R. Okay, this is the phenotypic ratio. Genotypic ratio is one is capital R, capital R. Okay, two of them are capital R, small r, and one of them is small r, small r. Okay, so what inference do you get? Still it is showing incomplete dominance, my dear children, and this is the phenotypic and genotypic ratio. You have to write like that, and this is a Punnett square. If you want to make the Punnett square instead of this, you can do that as well. Works. Chalo, next question. Give reasons for the following questions. So again, did I miss it? Oh my god, Jasmine. Okay, from here we are gonna start 5 marker question. Okay, we are gonna start 5 marker question, my dear children, supposedly. Okay, and yes, it is a 5 marker question. All of them carry 1 mark each, and you have to attempt 4 of them in your actual exam, but as of now all. So simple cut result in non-stop bleeding in hemophilia affected individuals. Why so? Why is that so? Because in hemophilia it is what? A disease, a genetic disorder, where the gene responsible for producing the clotting factor is mutated, my dear children. Now, if clotting factor is not there, who will clot? Hence, there will be non-stop bleeding. Cool. Let's read the answer first now. Non-stop bleeding occurs when a single protein that is a part of a cascade of proteins involved in clotting of the blood is affected. So obviously one protein in the whole cascade of protein is affected, but that is enough for non-stop bleeding. Now Turner syndrome affected females are usually sterile. That means they are not going to give rise to young ones, my dear children. Why do you think they are sterile? Because in Turner syndrome there is only one X chromosome, there is no other chromosome up here. So which is why their ovaries are rudimentary ovaries. Their ovaries are underdeveloped, or what do you call rudimentary ovary. So obviously they did not produced ovum over there. So this is the answer. Again, let's read. Females are usually sterile because they have only one X chromosome, leading to underdeveloped ovaries and lack of normal gamete production, just like what I said. Okay, now next thing. In Morgan's dihybrid cross experiments on Drosophila showed that flies having genes for yellow body and white eyes exhibited less recombination. So if I am talking about genes, I am saying they are showing less recombination. That means these genes are very closely linked to each other. They are very close to each other. That is why recombination means basically I am cutting this part of the chromosome and putting it elsewhere, and that part comes over here. Whatever you get, whatever you do, these genes are going to be together only. So where is the recombination happening? So if there is less recombination, that means these genes are very tightly linked to each other. They are tightly linked genes, my dear children. That is why there is less recombination. If there is more recombination, that means they are loosely linked, they are far away from each other. So your answer is yellow body and white eyes genes in Drosophila show less recombination because they are located close to each other on the same chromosome, exhibiting genetic linkage. Okay. Now, inheritance of skin color in humans shows different phenotype. Why so? Why do you say different phenotype? In skin color, do we have same skin color? No, some people are very fair, some people are wheatish in color, some people are dark in color, but still every one of them are beautiful. Why? So, the beauty of everyone, that is not understandable, but yes, every one of them are beautiful. But why is there a difference in color shades, my dear children? Why do we have different phenotypes? Phenotypes means physical appearance. So, over here, understand that skin color which is there, it is a polygenic trait. That means multiple genes up over here are going to affect the skin color. Suppose this is not producing enough amount of protein, but this is producing enough amount of protein. But in me, this is producing enough amount of protein and this is not. So basically depending on that, since because multiple genes are affecting a particular character, this is why we all have different phenotypes. Okay. Now, this is an example of polygenic trait, as in multiple genes controlling one trait. So that is why it is happening. So Skin color in humans. It shows different phenotypes due to polygenic inheritance, where multiple genes contribute to the phenotype, leading to a range of skin color variations, different color changes. Okay. Now over here, accumulation of phenylalanine in the body of Phenylketonuria affected individuals. Why do you think that happens? Why is accumulation of phenylalanine happening, my dear children? So in PKU, that is Phenylketonuria, what is the thing is that this phenylalanine which is there, okay, it is actually getting accumulated instead of getting broken down. So usually phenylalanine is broken down to tyrosine with the help of an enzyme, that is phenylalanine hydroxylase. But in PKU, this enzyme itself is getting affected, which is why there is no conversion and which is why this thing is getting accumulated in the body. So your answer is what? Phenylketonuria, phenylalanine accumulates in the body due to defect in the enzyme phenylalanine hydroxylase, which converts phenylalanine to tyrosine, causing the toxic buildup. Okay, there's a defect, which is why this is not happening. Okay, don't think this is happening, which is why the accumulation is happening. No, there's a defect, which is why phenylalanine is not getting broken down, which is why the accumulation is happening. Got it? Chalo, going ahead with the next question. Describe the steps involved in DNA fingerprinting technique. So again, I always give this example. For example, I am the culprit. I did a crime. I killed someone. I never killed anyone. Okay. So basically, and they found my blood over there. They don't know it's my blood. I have a friend of mine with me, my best friend. Okay. So over here, that blood sample was taken. Okay. The blood sample will obviously have cells. And the cells, there is DNA. So they had the DNA. What did they do was, they made a complementary strand out of it. They found a complementary strand out of it. Now, this is going to be known as a probe. What is it? It is a probe, my dear children. The complementary strand. Okay? You will get to know why. Now, I and my friend, both of our DNA is extracted. Isolation of DNA has been done. Okay? From the sample, not ours, from the sample. Also ours also are done. Okay, now digestion by restriction enzymes. Our DNA is being cut using the restriction enzymes, and basically after that it is separated using gel electrophoresis. So there is clear separation. Then from the gel electrophoresis chamber, things have been transferred to a nitrocellulose sheet like this. So everything is up over here. Now what do? Basically, suppose this is my. Okay, similarly I have the DNA as well, right? The DNA is also here. Suppose when the DNA was cut and segregated, it was showing a band like this. Okay, and now when they took mine, this is mine and this is my friend's. So when they took my band, it appeared like this. When they took my friend's band, it appeared like. Who is similar? This and this, obviously. Now that is a reason. And also I have the probe with me to confirm. What do you do on the nitrocellulose sheet? You send this probe. This probe will go and attach, and this probe, my dear children, is radio labeled. So this will go and attach to the complementary strand and will be lightening up under the UV radiation and will be showing that, hey, this is the complementary strand, and then it's pakka. Now that is known as DNA fingerprinting technique, my dear children, because we all have unique repetitive sequences in our body which no one else has. So that is acting as a fingerprint. So we are similar. 99% of our DNA is similar, but there is change in 0.some% of our DNA, and that change is because of the repetitive sequences which are different in each of our body. And these, with the help of these repetitive sequences, we can find things out. So these are the steps involved in the DNA fingerprinting technique, my dear children. Hybridization with the DNA probes, as in you're putting the DNA probe over here and you're visualizing it under X-rays or UV rays. Okay, and obviously you can compare like this also. Fine. Chalo. Next thing, name the causative agents of the following diseases. Malaria. What is that thing that causes malaria? Easy question it is. Okay, malaria is caused by Plasmodium. What Plasmodium? What is the most malignant version of malaria? It is caused by Falciparum. Sorry. And then you have Vivax also. Either way. Now, or Plasmodium malaria. Now, filariasis is caused by the filarial worm, which is known as Wuchereria bancrofti. Wuchereria. please make sure you're writing the species name in small letters and this one in capital letters. Okay, Bancrofti or you have Eukaryaria malai, either one. Now Ascariasis is caused by the roundworm which is known as Ascaris lumbricoids, but you just have to write Ascaris, that is fine because in your NCERT it's not there. Amoebiasis is caused by Antamoeba histolytica. Antamoeba histolytica. Talking about pneumonia, pneumonia is caused by Streptococcus pneumonia or Haemophilus influenzae, either way. Cool? So that becomes your answer for this particular question. Okay, next. With respect to the microbial products, its source and uses, identify A, B and C in the following table. So cyclosporin is produced by A, which is acting as an immunosuppressant. What do you mean by immunosuppressant? The one that has capacity to suppress our immune system. Why? Because probably I took organ from other human being because mine was not working. So at this point of time, my body should not feel that it is foreign and should not kill the organ which is there. Now that's why I need immunosuppressants. So cyclosporin A is produced by Trichoderma, my dear children. Trichoderma polysporum, remember biocontrol agent, it is also acting, it is also going to produce this polysporum. Now next, Monascus purpureus produces something that lowers the blood cholesterol level. Now, Monascus purpureus is producing what that lowers the blood cholesterol level, my dear children? It is producing statins. Now, statin stabilizes the blood cholesterol level. Okay. Talking about the next thing, streptokinase. Streptokinase is an enzyme that is produced by Streptococcus bacteria. Now, what is the use of this? Streptokinase is used for acting like a clot buster. So, whenever we have clot in our body, to bust that up, we have streptokinase. Okay, cool. And that is from microbes in human welfare. Now, you have defined BOD and Flocks. What do you mean by BOD? BOD is biochemical oxygen demand. So, for example, I'll tell you a story. There's an anaerobic bacteria, there's an aerobic bacteria, okay, whose name is PUC. Now, PUC says, I need oxygen to break down organic matter. PUC says, I need oxygen to break down organic matter. So, at this point of time, the biochemical oxygen demand is high for oxygen. Now, suppose PUC broke down so much organic matter. At this point of time, obviously, the biochemical oxygen demand will be less because there is no much organic matter left to be broken down. So, to check whether the area is polluted or not, what do? You test the BOD. If the biochemical oxygen demand is way too much, that means there is too much amount of pollution, which is why PUC is saying that I have high demand for oxygen. Okay. Now, suppose if it is less, that means there is less organic matter. That means PUC have already broke down. Now, you do not need much amount of oxygen over there. Okay. Now, what is Flocks, my dear children? So, when you do sewage treatment, right? So, you put the organic matter and everything into a tank, that is aeration tank, where you add aerobic bacteria, okay, to break down this organic matter. Now, the aerobic bacteria teams up with fungal filaments and all. And they form a mesh-like structure that is known as Flocks. Which is rich in this bacteria and fungal filaments which breaks down the organic matter. So, that's a good thing. Okay. Now, over here. So, cyclosporin A, statin, I told you everything. BOD is the amount of oxygen required by microorganisms to decompose the organic matter present in the given water sample, in 1 liter water sample, something like that. Okay. So, please write that. Now, Flocks are clumps or clusters of fungal filaments as well as bacterial cells and other materials that are held together by extracellular substances. Extracellular as in outside the cell. Okay. Extracellular substances produced by the microorganisms. Now, these aggregates are involved in biodegradation of organic matter during processes like wastewater treatment. Okay, I already explained you. This is the answer. Now, 41st question. Mention the population interaction that exist among the following: abundant tortoise and goats. So, you know what? Abundant tortoise were eating the grass and everything. Then goat came. Goat said that, hey, I also want grass. Please move aside a bit. And the goat started eating the grass. But what happened was that goat was the only thing that ate the grass. The abundant tortoise didn't have grass to eat, which is why they had to they moved out of it. They completely showed something that is known as competitive exclusion. So this is an example of competition, my dear children. This is an example of competition. What about tiger and deer? Tiger does not go and love the deer, right? They eat it. So this is an example of prey-predator relation. Prey-predator, where who is the prey? Tiger, and deer is the predator. Prey is what? The deer and tiger is the predator. Now sea anemone and clownfish, they have a very cute relation. Have you seen sea anemone or clownfish? Have you seen? You know, what's Finding Nemo? In that the Nemo fish which is there, that is the clownfish. Sea anemone and clownfish basically have a really good relation which is known as mutualism. They kind of help each other. Okay. Now wasp laying egg in the fig fruit, my dear children, they again show mutualism. Okay, where the wasp is laying the egg, providing a safe space to the fig plant, and the fig plant is basically able to do the pollination with the help of wasp coming every single time. Now cuscuta growing on the hedge plant. Have you seen cuscuta? Cuscuta is the yellow color thing that you might see on plants. Okay, they do not have chlorophyll of their own, so they basically go and depend on the plant for chlorophyll, and it's not a nice way of doing it. They literally get inside the plant with the help of a structure that is known as hostoria. Okay, with a structure that is known as hostoria, and take the water and nutrients and everything from the plant. This is known as parasitism. So Abingdon tortoise and goats competition, tiger and deer parasitism, sea anemone and clownfish mutualism, and what? Wasp laying eggs in the fig fruit mutualism, cuscuta growing on the hedge plant, again parasitism. I don't know why is question number given as 12 over here. It is 14th. 41. 41, and with that I think we have completed the 5-marker question where you have to choose 4 of them. Now we have in part D only, this is part D, right? In part D you have section 2 where you have to choose one question among next 3 5-markers. Okay, so I will be talking about that. Just give me 1 minute break. I'll just quickly drink some water and come. Okay. Okay, let's get ahead with the next question. Now, as I said, in the 3 questions that I'm going to show you now, you have to attend only 1 during the exam. Okay. So picture of a mature angiosperm embryo sac is given below, and answer the following question that is there. Which cells or nuclei of embryo sac produce zygote and primary endosperm nucleus? Again, let's hear a story. So when the pollen grain comes, no, it brings 2 male gametes. It brings 2 male gametes. One male gamete will be like, X cell is mine, I will be fusing with the X cell. So one male gamete fuses with one male gamete, my dear children. How much DNA, how much chromosome do they have? It's a gamete. It's a gamete, so gamete always have half the number of chromosomes. So it is haploid. One male gamete fuses with one female gamete, that is the egg cell, my dear children. It also has half, that is haploid. Together they give rise to zygote. And N comes together, it becomes diploid. This process is known as syngamy. This process is known as syngamy. Another male gamete does not have any option. So basically it goes and fuses with the 2 polar nuclei right over here. There are 2 polar nuclei that you see, my dear children. So the next, second male gamete, it's not 2 male gamete, okay. The second male gamete fuses with 2 polar nuclei. I'm so scared to put 2 over here because you might remember it like this. So I'll write second. Second male gamete plus 2 polar nuclei. Now here also male gamete is haploid. 2 polar nuclei, there are 2 of them. So each of them are N and N. So N. Now this is going to produce something that is triploid. And this triploid thing is what? Triploid endosperm, my dear children. Okay, or you also call it primary endosperm nucleus. Primary endosperm nucleus, if you talk just about the nucleus. But you know, this polar nuclei is a part of a bigger cell which is known as central cell, my dear children. Okay, central cell. So basically it is forming primary endosperm cell. Either way. Now this process is known as triple fusion. Why? Because 3 things are fusing together. And which is why you call it double fertilization. This is one fertilization, this is second, double fertilization. Now, which cells nuclei of embryo sac produce zygote? You know, it is these cells. you're asked about of the embryo sac. It is a female gamete, that is an egg cell. And what about primary endosperm nucleus? It is a two polar nuclei, or you can say it is the central cell. Please be sure about what is asked in the question. You're asked about embryo sac, so please mention about that. Now, what is the ploidy of antibodal cells and the primary endosperm nucleus? You saw the primary endosperm nucleus, my dear children, it is triploid. N. It is what? Triploid in nature. So, this is triploid. Okay. What about antibodal cells? So, this is egg cell. Egg cell has two best friends, synergids, which will be helping the male gamete to come over here. Now, this is the central cell having the two polar nuclei. Oh my god, sorry, this is the egg cell. Okay, sorry. This is the egg cell. These are the two synergids. This is a filiform apparatus through which the egg cell will be coming in. So, sorry, the male gamete will be coming in, okay. And the opposite end, you have three antipodal cells. Usually the picture is like this, which is why I got confused. So, you have three antipodal cells. All the cells, my dear children, which you see over here, they would be having haploid amount of chromosome. So, it is N. So, all the here also the ploidy of antibodal cells is what? Haploid. Don't take it N, that is triploid. No, separately you are asked. So, it is haploid, and the primary endosperm nucleus which is there, it is triploid for sure. Now, why the endosperm development precedes the embryo development? You know what? The endosperm, primary endosperm nucleus, no, it divides and give rise to the endosperm tissue later on. Why do you think endosperm develops first, much before the zygote? Because this endosperm is the one that has to provide nourishment to the zygote to become an embryo. So, initial nourishment is provided by the endosperm itself, which is why it's preceding development. Got it? Oh, okay. Chalo. Next question. Answer the following. Construct a complete transcription unit with a promoter and terminator on the basis of hypothetical template strand. This is a template strand, my dear children. So, now over here, listen. This is a DNA. In DNA only you have template strand and this strand and that strand. Okay. So, it is mentioned like this. When the arrow goes like this, that means this is 3' end and this is the 5' end. Opposite will be 5' end and this will be 3' end. Cool. Now, over here I have to write all this. A, T, G, C, A, T, G, C, A, T, A, C. Cool. Now, here this is the template strand which is going to undergo transcription. The opposite strand is known as coding strand. It will have the complementary base pair, that is A with T, here A, G with C, C with G, A with T, T with A, G with C, C with G, A with T, T with A with T and C with G, right? This is the coding strand. Now, over here, my dear children, you will be having the promoter region for transcription to happen and you will be having the terminator region as well. Always remember that. Now, this is a transcription unit. Now, write the RNA strand transcribed from the above transcription along with the polarity. So, this is the strand that is going to get transcribed, and always remember transcription happens from 5' to 3'. That does not mean this is getting transcribed. No, from 5' to 3', that means this is going to get transcribed. That's when the complementary strand will be in 5' to 3'. So, what's the easy way to do is, you have the coding strand, just replace T with U, because in RNA you do not have T, you have uracil. So, it will be UACGUACGUAUG. That's all. And this will be with polarity 5' to 3'. Now, the problem comes when you have messed around the coding strand. Okay, you didn't write the coding strand properly, then obviously you won't be writing this also properly. So, make sure you're making just the complementary ones there. Okay, so this is the transcription unit. This will be how it looks, my dear children, with promoter and terminator region 3' to 5'. That will be the template strand. Please mention the template strand as well. Forgot to mention it. And this is the coding strand, and this is the mRNA transcript. Works? Chalo. Next, don't forget this. Okay. The last question of the day. Study the diagram given below and answer the questions that follows. What is EcoR1? EcoR1 is a restriction enzyme, my dear children. Okay, which is obtained from Escherichia coli, and it is known as the R strain, and 1 is because it was in the order of discovery, it was produced 1, 2, 3 and things like that. So, EcoR1 is a restriction enzyme, is the simplest answer that you can give. Now, have I mentioned the answers up here? Yes. So, Econ R1 is an estrogen endonuclease. What do you mean by endonuclease? The one that cuts within the DNA, that is endonuclease. Enzyme derived from Escherichia coli. It is recognized the palindromic sequence 5GAA-TTC. This is the sequence that it recognizes, and it cuts between G and A, and cuts the DNA specific site, producing what? Sticky ends. So in one strand probably it cut between this, another strand that has complementary thing. So here the complementary strand will be like GAA-TTC, right? So here also it will make a cut. So there are two sticky ends that is going to be produced. Over here, if you do not understand, I can show you like this. So this is GAA-TTC, GAA-TTC. Here it made a cut over here and here, right? So see, you can see now, yes. So I made a cut over here and here. What did I get? I get the whole strand removed up over here. Similarly, in other strand also, I made a cut over here and here. What did I get? I get a whole strand over here. So these are sticky ends. This is a sticky end, this is a sticky end. Okay, since if you see they have complementary base pair, T will be binding with A, T will be binding with A will be binding with T, and A will be binding with T. Get it? So if I make them close to each other, they are going to bind with each other. You get it? So these are sticky ends, and you get it when you are going to cut it with the same restriction enzyme, my dear children. Okay, we are going to get that question as well, but yes, let's focus on this. Now, over here, how is the action? Oh, so you have to remember the site. Okay, what is the site? My hand is paining. 5-GAA, GA TTC. You can remember like that. GA TTC. Okay, GA, give me TC. No, GA TTC. That's the best way to remember. Okay, so remember 1, 2, 3, 4, 5, 6 nucleotides are there over here. Now, how is the action of exonuclease different from that of endonuclease? Exonuclease are enzymes that would be cutting the DNA at the ends. Endonuclease are enzymes that will be cutting the DNA from the middle part, and they do it by reading the whole DNA sequence and everything. It's not a random process that happens. It is very specific. So difference is exonuclease removes nucleotide from the ends of the DNA strand, works in a sequential manner from either 5' to 3' end, 5' or 3' end. Now exonuclease cut within the DNA strand at specific recognition sites, creates either blunt or sticky ends, my dear little children. Okay, now over here, how are sticky ends formed on a DNA strand? Why are they so called? So why do you think the sticky ends are being formed, my dear? Okay, why do you think sticky ends are being formed? As I said, we need to join two sets of DNA, right? So from here, this is a vector DNA, okay, and this is probably the thing where you are going to put it. Probably, what do you say? The DNA that you, foreign DNA that you want to put it. Okay, so both of them you are going to cut like that, and you are going to make them come closer together with the help of sticky ends. So why are they so called sticky ends? Because they do not have a complementary base pair, so they can easily make hydrogen bonds with anything that is complementary to them. So how do you write it? Formation of sticky ends and why they are called so. So sticky ends are found when restriction enzyme, that is example EcoR1, cut the DNA specific palindromic sequence. Palindromic as in the one that can be read in both the direction, it will be read the same. So GAA-TTC, here also if you read GAA-TTC will be the thing in the complementary strand. Okay, leaving single stranded overhangs. So it will be leaving single stranded overhangs, as in you saw over here, they are single stranded and they are overhangs. Okay, now these overhangs can be easily bind with complementary sequences as well, and they are called sticky because they have exposed bases that can form hydrogen bonds with the complementary DNA fragments, facilitating the process of recombination. Okay, they are known as sticky. Why? Because they have exposed bases that can form hydrogen bonds, and with this we are completing all the questions of model paper 2. I hope that everyone who watched this particular video have understood each and every question, but still if you have any doubts, please feel very free to ask me. I'm not here to judge you, I always say that. So just ask the questions, I would be too happy. And even if you like the video, you have to tell me, ma'am, I understood everything. So you don't have to tell me, ma'am, you taught good and things like that. Just say, ma'am, I understood everything. That is more than enough for me. Okay, and also please do not forget to ask your doubts. I'm here for that. So that's all. Please do not worry. You're gonna do very, very good in your exam. Bye-bye. Take care to all my cute little champions.